Okay, well, Dan, thanks again for joining us. I wanted to share a bit for the group, your background. Dan Mason is the leader of the eDNA assay development team at the National Genomics Center for Wildlife and Fish Conservation. As a graduate student, he studied the ecology and genetic evolution of several freshwater species of conservation concern and joined the Rocky Mountain Research Station in 2017 after earning a master's in science. Since then, he's provided laboratory and field support to the development of eDNA assays and methods for more than 50 species, both aquatic and terrestrial ecosystems. Some highlighted projects for 2022 include sampling the Colorado River from Lake Powell to Lake Mead to survey native and invasive fish communities, developing eDNA sampling methods for endangered pocket gophers, and developing eDNA protocols for detecting snow leopards in Central Asia. And I'm sure that is going to be a fun field trip when you get to go look for snow leopards, right?
Yeah, if I'm lucky, I'll be the one who goes, but there's going to be people ahead of me, maybe.
Yeah. Well, it sounds like you've done some fantastic research, and we're looking forward to hearing your talk tonight. So go ahead and share your screen.
Great. How's that looking?
Looks great.
All right. So yeah, hello everyone, and thank you all very much for letting me talk to you today about environmental DNA, which you're going to hear me refer to as eDNA most of the time. First, I want to explain what the National Genomics Center is and what we do. Then I want to talk about how eDNA methods work and what we frequently use them for. And then I think I'm going to wrap up with a look at some eDNA results from the Hood Watershed. But this is kind of an unstructured talk. So yeah, stay on your toes. And aside from being unstructured, it's going to be a pretty general coverage of the material, because I think for some of you, this might be the first eDNA talk you've attended. And also because I'm not a Hood River expert, and all of you are Hood River experts. So I plan to cover this material well enough for all the experts in this meeting to start to think about how it applies to the interests and to the mission of the Watershed Group. All right, so first off, the NGC. The Genomics Center is located in Missoula, Montana, and is part of the Forest Service Rocky Mountain Research Station. The Forest Service is known by most of the public as a land management agency, and rightly so, but it does also contain an R&D branch, and that branch is divided regionally into research stations. So our station focuses a lot on the Rockies, but we do have partners in other parts of the country. And so our purpose in the most general sense is to provide scientific leadership to the Forest Service, the national forests and the Forest Service regions, and to Forest Service partners as they design and implement projects to manage public lands, public waters, and of course the species that those lands and waters support. So starting with the National Link Survey in 1999, which some of you might have been around for, the National Genomics Center has been responsible for providing the science leadership in the application of genetic and genomic methods to fish and wildlife research. And broadly speaking, one of the most common reasons why we're asked to supply this type of support is to help deal with what I kind of call the rare species problem. And so what I mean by the rare species problem is that rare species are often some of the ones we're the most eager to find, the ones that we work hardest at trying to observe. And despite being super interested and working really hard, we still are sort of bad at finding them. And since it's so hard to find them, surveys for these kinds of species take a lot of time and they take a lot of resources. And as a result, many of the species are only ever surveyed in a small portion of their range, or they're surveyed infrequently, like, once every 10 years or so. In some cases, it can be both of these things. Only infrequent surveys are done in small areas. And what that really means from a conservation standpoint is that potentially huge changes can take place and go unnoticed for long periods of time. And that makes management and conservation a lot more difficult and expensive when you're kind of behind the 8 ball, so to speak. So since about 2015, the Genomics Center has used eDNA methods to circumvent this type of problem for aquatic species of concern. And essentially, eDNA is DNA that is shed from the cells of an animal and deposited into its environment. This could be skin cells, it could be saliva, mucus, urine, feces, basically anything gross that falls out of or off of an animal. And we can kind of follow in their footsteps, so to speak, and recover this DNA from media like stream water, sediments in river bottoms or lakes, dirt, snow, and occasionally we can even get it from air. So this kind of schematic here, we have a fish that's being spied on by a snorkeler in this picture, but it's also shedding DNA into the stream, which we can pump through a very small pore size filter and then take back to the laboratory, run an eDNA analysis, and kind of map our detections or non-detections onto the landscape. And also, I just realized, I'm not sure if you guys can see my cursor or not.
Yeah, we can.
Okay, great. I was waving it all over the place, and yeah, if it gets annoying, let me know. So yeah, once we have an eDNA sample, we can use what we call an eDNA assay to detect a particular species of interest by isolating and amplifying that species' DNA and only the DNA from that species, not any of the other species that are present in the sample. So these assays work using similar technology to another type of assay, which you're actually probably all pretty familiar with by now, and that is the COVID PCR tests. These assays rely on a technology called quantitative PCR or qPCR, and it's extremely sensitive. An eDNA assay can detect as few as a single copy of a DNA fragment, just one little piece in a reaction, and it's, I think, statistically pretty hard to get better than that. In a more applied context, eDNA methods can detect a single fish about the size of your pointer finger about 85% of the time if the sample is located within 100 meters. So for detecting population level occurrences of fish, we have more than enough sensitivity to do that for sure. There's also some experimental work that's been done that shows eDNA from some of these aquatic species can be detected at least as far as 12 kilometers downstream. So you don't have to be 100 meters, so to speak, from your fish to be able to detect it. It's just that the closer you are to it, the higher your probability of detecting it will be. In any case, if you have an eDNA assay and a sample, you can really accurately identify which species were present in the area around the time that the sample was collected. So in a simplified form here, this at the top is a qPCR plate being loaded in a stock photo by somebody who should really be wearing lab gloves. And we have two outcomes possible here, which is The left one is an amplification shown by this spiked curve, and the right one is a non-amplification shown by this flat curve. So this one on the left, we would call the species detected, and the one on the right, we would call it not detected. And I also want to point out that these samples can be collected really cheaply. They can be collected by one person on their own with pretty minimal equipment. This picture shows you everything you need to go eDNA sampling. And actually, if you were going to do a backpack sampling trip, you might even trim some of this out as just being not necessary. And it doesn't take a whole lot of training to be able to do it really effectively, as much as I would like to brag about my skills. In sampling with eDNA, I think our youngest field volunteer so far was 12 years old, and we didn't have to have any like uncomfortable conversations about their job performance, which was good because they're my boss's favorite kid, actually, and that would be awkward. eDNA does still have some limitations. It can't provide us with absolute abundance information. It can't give you a number of fish counted, and it can't identify specific individual fish. Like this is fish 0001 from 2016 or something like that. It can provide relative abundance information, as in there are more of this fish here than there are here. And we have other genetic methods that we can use for identifying individual fish by kind of recovering their unique genotype from a DNA sample like a fin clip or a swab. So essentially, eDNA excels in two extremely useful ways. Actually, it's more than that, more than two, but I'll talk about the others at the end. It is fast and cheap enough, though, to use to sample really large areas, like the size of a state or the Rocky Mountains, for example. And it's sensitive enough to detect what you're looking for, even if there's only one or two individuals from that species present. So when we can take these really comprehensive surveys that have high sensitivity and put them into context, we can make really informed decisions about how we manage. And I'll be able to switch here. The way that we are currently putting eDNA results into context is through the use of the Aquatic eDNA Atlas. So that is the map looking thing you see on the screen now. And this is an ArcGIS online platform that provides a pretty convenient way for people outside of our lab to view results from our lab spatially. So here I have the Hood Watershed. You should hopefully be able to see the black points if I untoggle that there. So these are sites that we've collected an eDNA sample from. We, in a broad sense, like our partners and ourselves. And if I toggle the GIS layer that contains results from a specific assay I'm interested in, I can display species detections on top of this base map. So here I've selected bull trout, and the blue points show locations where bull trout were detected, and the green ones show locations where we did not detect bull trout. Sites that are still black haven't had their samples analyzed yet. So we can come back to this map later for people who are curious about exactly where all these points are. But bull trout are one of those rare species which previously took a huge amount of effort to find. And once we had designed a bull trout eDNA assay, we could easily find them, much more easily, and work with our partners to pretty much completely survey Washington, Oregon, Idaho, and Montana for bull trout. So this is the same resource, but backed out a bit, and it shows locations where we have surveyed for and detected bull trout using an eDNA assay. And basically, we now know where bull trout are and are not present at 1 kilometer spatial intervals on any of the national forests in those 4 states that have bull trout range. And now that we know where bull trout are, we can start to better understand how to go about conserving them. Bull trout have both a resident and an anadromous life history. They're sensitive to water quality changes and stream disturbances, and they're cold water adapted species. And this last characteristic, I think, is going to present an enduring challenge to bull trout viability in the U.S. going forward. So that leads me to a disclaimer. And the disclaimer is I am not a climate scientist by any stretch of the imagination. I do, however, work with a few climate scientists, so I get to use their stuff in my talks. And this is a forecasting tool produced from an effort led by Dan Isaac out of Boise and Mike Young, who's based here in Missoula, called Cold Water Climate Shield. And I should show you the actual tool instead of just the paper. So here we have the hood again. And the purpose of this tool is to predict the probability that a stream will be able to support rearing of a cold adapted species. In this case, it's bull trout across a couple of different potential future climate scenarios. So just as a reference, this is what Climate Shield predicts for a climate scenario that's essentially the same as the climate was in 1980. And working downward through the legend here, dark blue represents a probability of occurrence greater than 0.9. Light blue is 0.75 up to 0.9. Dark green is 0.5 to 0.75. Light green is 0.25 up to 0.5. And yellow is less than 0.25. So the lighter the color, the lower the probability the model is predicting. And I'm not entirely sure if this is colorblind friendly, so I apologize if it's not visible to some folks. So now we can look at a different scenario, which is the probability of bull trout occurrence. And again, this is juvenile and juvenile bull trout, because we need reproduction to be viable. First scenario that is moderately warmer. The climate is moderately warmer. Perhaps something like what a trout might experience in the 2040s. So now in the hood proper, I see some here, but in the hood proper, there aren't any streams with a probability of 0.9 or larger. And I think only the headwaters of the Middle Fork have a probability of 0.75 or larger. And I do want to mention at this point, and I know there's a lot of map switching here, so bear with me, that much of the hood has been designated as critical bull trout habitat by the Fish and Wildlife Service. So this map shows in dark blue, I shouldn't have zoomed in because it's gonna have to re-render. In dark blue, this is critical bull trout habitat according to the Fish and Wildlife Service. And this designation means that maintaining habitat conditions appropriate for bull trout has been deemed essential for their long-term viability and should be a high priority. And I think this habitat work can be really extremely effective, but there are also no silver bullets. The principal reason for the development of Climate Shield was to predict future cold water refugia, which it definitely does do. But I think it also makes it clear that there are certainly going to be some difficult decisions to be made about which restoration projects we collectively ought to invest in as climate change kind of continues to move the goalposts and turn some of these quality habitats into more marginal ones. So it's my sort of pessimistic endpoint for bull trout for now. I'm a pessimist, I'm sorry. But not all species of concern are as cold adapted as bull trout, of course. The mountain streams of the West have been cold enough for long enough, though, that there are definitely other species we can apply this type of approach to. Cutthroat trout and Pacific lamprey are both fish that need cold water for rearing. Not quite as cold as bull trout like it, but still pretty dang cold. And both of these are species of conservation interest, especially lamprey, which are really deeply important to the indigenous peoples of the Pacific Northwest. And I just wanted to show some pictures of those two fish because they're both very, very nice and pretty looking, in my opinion. Before I go back to the atlas, and we're back to the hood now. So I can toggle Pacific lamprey results. And unfortunately, at this point anyways, we don't have an extremely exciting story in our results for Pacific lamprey detections because we haven't made any in the Hood watershed. We have detected them in the Bonneville Pool, and we've detected them in some of the other tributaries to the middle Columbia, like the Sandy River. I'm not entirely sure if this is the reason why. Maybe some of you have a better idea, but there are some types of fish passage structures installed on dams that are less effective for getting lamprey across than they are for bony fish like salmonids. So perhaps the removal of the Powerdale Dam, which I know this group was involved in, will give lamprey a better shot to recolonize the Hood than they had before. And yeah, so we can come back to this map and talk more specifically about where these points are. Both from a station perspective, a genomics center perspective, both of the bull trout and the Pacific lamprey projects are really large-scale efforts, range-wide surveys for these species. But one of the nice things about eDNA methods is that they can be really useful on small scales too. So we use eDNA tools for things like finding if a species is present above or below a culvert, or looking for any surviving invasive fish after a fish eradication effort has gone through and removed them. We use this in Montana a lot for brook trout. And it's really effective for that because we have, again, very, very high sensitivity. We can find one fish, even if it's the only one left. And eDNA has applications to surveys for other species as well, as in not just fish even. We use it really commonly to sample for frogs, salamanders, turtles, mussels, some aquatic pathogens. And we, in the past few years, have started to use it to survey for terrestrial mammals, including some forest carnivores like Canada lynx, wolverine, marten, fisher, cougar, bobcat, and red fox by doing winter track line surveys over snow. And so we hit a track and then we can scoop snow from the track and collect the DNA they have left behind in their footprints and identify the track line to species without any discrepancy, because track identification can be really error prone depending on snow conditions and experience. So really, I think for forested and mountainous watersheds like we have in Montana and like we have in the Cascades, eDNA methods can be applied with a lot of flexibility from an ecosystem management perspective. A lot of what I'm working on now is expanding the number of species that we have validated eDNA assays available for, and also looking for opportunities to apply new lab methods that will increase the efficiency of analysis, as in reduce the amount of the sample we need to use to get the data we want, and reduce the cost of the analysis as well. So aside from probably the obvious reasons, both of these objectives we think are really important because of the last strength of eDNA I want to mention. And that is, once the sample has been collected from the stream, it's essentially a time-stamped archive of the entire aquatic biota at that location, at least up until we've completely used the sample to analyze and search for species DNA. So we're keeping these samples in deep freeze storage indefinitely. And the more species that we have an eDNA assay for, and the more efficiently we can use each sample, the larger the amount of information we can potentially retrieve from these samples in the future. It's not just a one-stop shop kind of deal. So I think that wraps up how I wanted to kickstart this discussion. And I'm gonna throw some links in the chat for folks who want to check out these tools more on their own time. But I'd really like to hear if there are any questions or if you all have any ideas you want to share and discuss. So let's hear it.
Thank you, Dan. Do you want to stop screen sharing, or do you?
I can stop, and I can always start it back up again.
Perfect. Let's do that. That way we can kind of step back and see who's might have a question, and then you can easily go back to screen sharing if needed.
Sounds good.
And all right, thank you, Dan, for we see the those links. I've got a quick question to start things off then, and that is, you did mention that you can determine relative abundance. Can you describe that a little bit more?
Sure.
Yeah, how relative is relative?
Right. Yes. So that's definitely a question we get a lot. And so what I mean by relative abundance is kind of shown in this schematic, if you can see it. So here we have, on the left, we have brook trout detections, and on the right, we have cutthroat trout detections. And the size of the dot is related to the amount of DNA that we recovered from the sample. So I mentioned that the tool we use is called quantitative PCR, and it's called that because you can estimate the initial concentration of DNA in your sample or quantitate it. So if we run our samples with a reference curve of different concentrations, we can identify our unknown environmental sample concentration. And if you do that spatially in a kind of orderly way, then you can see patterns play out. So in this case, what we saw in Cooney Creek was we had higher concentrations of brook trout in the lower reach of the stream, and those concentrations dropped as we went upstream up towards the right-hand corner here, till eventually those crosses, we didn't get any brook trout DNA at those locations. Whereas for West Slope cutthroat trout, we see pretty much we see the inverse more or less, where we have less West Slope cutthroat trout DNA downstream, more in these upper middle reaches, and we have cutthroat trout much higher in the stream than we have brook trout. So we made the inference here, and we backed it up. We were actually doing brook trout removal, so we had electroshocking data. And the relative abundance of brook trout and cutthroat trout in our electroshocking data matched qualitatively what we were seeing in the eDNA results.
So if you get if you go out and do some field sampling to see like how many fish are in that, you know, that you could find like within that one of those circles, then could you say, you know, that size circle is roughly, you know, this many fish per quarter mile, or can you just not do that?
You can definitely try. I mean, that has been a goal of people in our field for as long as we've been using the method for fish. Most of the time you're giving up precision to the point where if you want to take action, you really want to do electrofishing surveys. If the density of fish in an absolute sense is critical to you, then eDNA is probably not the tool for that, because it turns into a correlation really, and you have a lot that's not known.
Okay, I think Dick had a question. Go ahead, Dick.
Yeah, I mean, this is a really fascinating subject to me. I'm a physical scientist rather than a biologist, but I find this really interesting. And a question I have is, so if you have a stream, and use the example you just used, where you have both cutthroat and brook trout, and I'm assuming that those two animals share a huge percentage of their genome, just like we humans share a huge percentage with chimpanzees. So how exactly, I mean, can you tell us a little, is it possible in simple terms to explain a little bit about the technology of how the machinery zooms in on finding, say, just the cutthroat DNA and then amplifying that so you can actually determine concentrations and so forth?
Sure, yeah. PCR stands for polymerase chain reaction. And basically what that means is it uses an enzyme called a polymerase to take a few copies of DNA and make a lot of copies of them. And DNA, as if you think back to kind of high school biology, it's stable in a double-stranded conformation. So the way that this technology works is we can, if we have DNA sequence data from brook trout and from cutthroat trout, we can design a really short piece of DNA that we call a primer, and we can make that sequence match whatever sequence we want it to. So it will be a perfect match, let's say, to brook trout DNA. And if we pick the right spot in their genome, it'll be a perfect match to brook trout in a place that it is not at all like cutthroat trout DNA.
So is it fair to say then that you need to know the genome of the species in advance so you can design that and then apply the technique? Is that true?
You don't need to know the genome. You don't have to know everything. You need to know, it can be as few as like 500 base pairs out of millions. So you have to have some information for sure, but it's pretty cheap compared to finding genome-wide information. It might cost you like $8, for example. Yeah, and then so once you designed a primer, you can create in the qPCR reaction, you can create conditions where the enzyme and the primers will only latch onto brook trout DNA, and they'll only make more copies of that. And as they make more copies, we can add another DNA molecule in there that gives off fluorescence when you shoot it with a laser. And that fluorescence rises in proportion to the number of copies. So that's what generates that curve that I talked about. That spike curve is actually fluorescence we're reading off the machine, which represents more copies which have been made. And the flat curve represents no increase in fluorescence. Does that answer your question?
Yeah, it does. And I find it spectacular that, that technologies, it has advanced to the point where this is a really practical, useful tool. It's really quite impressive.
Yeah, I agree with you. And I remember, I think even when I was in school, the people were really just starting to tinker with this for environmental samples. And that wasn't that long ago. And now it's big scale projects type applications. Yeah.
Dan, you want to go? Dan Ball, do you want to ask your question?
Okay. Yeah. Hi, Dan Ball here. Can you guys hear me? So Daniel, I'm just curious, has this technique been used or developed at all for aquatic vegetation or riparian vegetation? I'm wondering if it has potential for, aquatic weed identification and things like that.
Yeah, it has a little bit. I think it hasn't been used quite as much because of funding and priority reasons rather than technical reasons. I do think it's been used for Eurasian milfoil, which is an invasive plant we've got issues with in Montana. And certainly from a biological perspective, I don't think there's a broad reason that I could say it wouldn't work really well.
Sure.
Plants have slightly different genomic makeups than most vertebrate animals do, but people do plant genetics all the time. And yeah, I think we know how to do it for sure.
So the potential would be there for that kind of surveying, such as you've used for these other species.
Yeah, for sure.
Cool.
I think it's very uncommon that the genetic or the genomic problems prevent an assay from being possible. It's more common that maybe we thought filtering stream water for this species would be really effective, but it turns out there's not a lot of its DNA in the stream for whatever reason. So I mentioned that we do use this tool for turtles, but turtles have a super low metabolism. They're not giving off a lot of waste products. They don't have a lot of epithelial cells exposed to their environment. So compared to like a trout, a turtle gives off way fewer copies of DNA compared to that trout. And you have to be a lot closer to the turtle to pick it up than you do to the trout, so to speak.
And aquatic plants don't poop quite that much.
No, they don't. They do disperse. They depend on water transport, a lot of them, for their dispersal though. So they release seeds, or they themselves are carried in the current. And that would, if I would make a guess, that would be probably how we'd capture DNA from them. It'd be less like microscopic cells and more like chunks of plant.
Thank you.
Mm-hmm.
How about mussels? Do you know their relative sort of conduciveness to this kind of sampling?
Yeah, it works really well for mussels in our experience. We're doing a lot of mussel analyses right now, actually, for Western ridge mussel, because I think the state of Idaho is trying to figure out where they do and don't have those. But yeah, we've done California and Oregon floater. We've done Western pearl shell. There's another one that's on the tip of my tongue that I can't remember. Of course, we don't do a lot of analyses for this, but there are other labs that do a lot of dracinid mussel analyses in the Midwest because they're looking for invasive mussels. And yeah, it seems like it's a very appropriate tool to look for those critters.
I have a question about the lamprey data.
Yeah.
And I admit that I missed the last watershed group presentation about lamprey, but I'm hoping that other people here know more about the status of lamprey in the Hood River. I feel like maybe a couple of years ago, I heard some presentation or somebody's data about lamprey surveys and saw a map at one of these presentations of all the places that they had been found. So I was wondering if that's like, if the difference between finding them in past surveys and them not showing up in this eDNA sampling is just a matter of timing, or if there's another explanation for that, or maybe they weren't actually Pacific lamprey.
Yeah, actually, I was gonna ask if they were Pacific lamprey.
I can probably answer that. So just based off your map, and the tribes have a lamprey specific project in the Dalles that looks at 15 Mile Creek, Hood River, Lower Deschutes, and the lamprey are right now below the Forest Service, below where you've sampled. So they're up the East Fork, at least past the East Fork diversion. They're in the lower West Fork now. They found amicetes. They've actually found individual,
Great.
with electrofishing. It looks like you have some samples that aren't done yet at the lower Hood River. You're going to find lamprey there. I've seen them myself, those are going to show lamprey. So I think the distribution is just below where the Forest Service lands are right now.
Well, it's good to hear they're coming up, or potentially coming up.
Yeah, and you mentioned Powerdale Dam, and that was not a good, that was not effective for lamprey passage, and they've been working their way up. So it looks like they've got above Punch Bowl Falls on the West Fork, so that's a, kind of a potential barrier. But they seem to have got up there. So we'll see. Like if you continue this, it'd be really interesting how far they make it up, or how that range changes.
Yeah. The initial stage of the range-wide lamprey project was designed to sample pretty coarsely to identify, like, is this tributary occupied or not, so to speak. And then the subsequent stage will be going back and sampling at a smaller spatial scale. Or where we had detections at least. So yeah, hopefully that type of thing is something we'll see a lot in other streams outside the Hood as well.
I have a question for, I've got a question for the group, some of the partners here, whether you could see using this technology, because Ryan, I know, we've been talking about, you've expressed an interest in doing more of this, and Kat, you're planning on continuing this as well, I believe. So what about something like summer steelhead spawning in the West Fork, where it's hard to, it's probably hard to pull off a spawning survey when summer steelhead might be spawning, but it would be interesting to know, potentially, are we thinking about something like that for use for eDNA sampling?
So I would like to kind of expand on that. And I guess this question is for Daniel. It's a little bit about feasibility. So, if we have two stocks of steelhead, we have summer and winter. They're the same species. And you said you can't tell individuals.
Mm-hmm.
Do you have kind of any sense, could you, summer and winter steelhead are genetically distinguishable. There are markers for run timing and all this, but is that too fine a detail to pick up the difference between a summer and a winter steelhead? Do you think that's possible? Or are they going to kind of all show up as an omicus? Can you tell different cutthroat subspecies apart, for instance?
So answering backwards, yes, we can tell different cutthroat subspecies apart. But no, we can't tell different timing of the runs apart for steelhead. The run timing markers that I think you're referring to rely on genomic scale data. So they need thousands and thousands of markers to pick out the single base pair differences that are represented in those two different populations. And I think the last time I looked at it, there's only 7 or 8 differences that are absolute, like they're only found in one of the runs and not the other. And they're spread out hundreds of thousands of base pairs apart from each other. And that type of distribution of genetic distance in the genome is not something that we can use qPCR technology to pick up, unfortunately.
That's kind of what I thought and was worried about, but yeah, that makes sense.
Yeah, someone asked about it a few years back, and I wanted so badly for it to work, and it would. Yeah, I don't think it will.
Hey, Greg, I'm sorry, you had your hand up. Go ahead and ask your question, Greg Short.
Yeah, I was curious if the same technique or technology would work for certain mammals or birds that spend a good part of their life in the water, if that would work, if it's used for that as well.
Yeah, we have used it for that in the past. We've done harlequin ducks with Glacier National Park, and we were able to detect them with eDNA, but harlequin ducks are also fairly easy to detect with your eyeballs. So
yeah,
we didn't see a huge benefit to really using the method a lot. We've also done beaver and river otter for mammals, semi-aquatic mammals. And it's kind of a similar story as the turtle example I mentioned. Since they don't spend every waking second in the water, it does seem like you have to be closer to have the same probability of detecting as we've kind of gotten accustomed to with trout, for example. We're not sure yet in the long term if it's going to be a higher probability than actually going out and doing an otter survey. It certainly might be less effort if you don't have to set traps, or you don't have to set camera traps. But yeah, that's something we're interested in doing.
It might be more economical in the future if the sampling gets even less expensive with technology than actually going out and doing the hours and hours of work on the ground or in the river or whatever.
Yeah, for sure. Yeah, I think the cases that we've done it so far have mostly been projects that were funded for other reasons, and then for various reasons, the money, there's more money left over in the pot when the project is done than we thought there would be. And so they say something like, hey, it'd be great if you could rerun our samples for otters or beavers, just because we think we have them, but we don't know exactly where they are, that kind of thing. So it's more of an add-on. We haven't had a full-blown, like, let's find otters across the Rockies project yet.
Any other questions from anyone? Okay, I'm not seeing any hands or voices. So I think at this point, we really need to give a round of applause and thank you to Daniel for what is a great presentation. Thanks a lot.
Yeah, thank you all very much.
I'm sure